Hello students, today we are going to start with a new chapter, that is biotechnology and its application. In the last chapter already we have discussed the principles, how actually biotechnology works and how we can able to do with the help of the biotechnology. So in this chapter, biotechnology and its application, we will just see how many types of products are created in different fields and how it is helpful for human being. All these things, the applications basically, we will see in this chapter. The procedure we are not going to discuss because the procedure we have already discussed. So in this application we will see only the products. So first of all, we should know actually that biotechnology help in how many ways, in how many different fields. So first of all, just see what is the scope of biotechnology. That biotechnology can be used in different fields, right? So see, first of all, this biotechnology is used to develop the GMO. So we have to know the first thing, that what is the term GMO, that abbreviation GMO indicates. GMO stands for genetically modified organisms. So genetically modified organism is the most important product of biotechnology, which is helpful for human being. Actually it is created, it's manipulated for the human use. So that is the GMO for the better yield, therapeutics, then sewage treatment, energy production in different scopes. These different scopes of biotechnology we have already seen in the earlier chapter, that the sewage treatment, in the sewage treatment also biotechnology is used. We have seen that the different bacteria, anaerobic bacteria as well as the aerobic bacteria with the fungi help in decreasing the pollutants, organic pollutants from the water and make the water usable. For the production of energy, that means biogas also, biotechnology is used. That means gobar gas or that cow dung, from the cow dung what we can extract the gas that is useful, that can be used in the different uses, like in the kitchen also, for the production of light also. Biotechnology is helpful in production of energy. So it is helpful for the sustainable development, that we have created some alternate source of energy without hampering the fossil fuels. So this way biotechnology is useful in lots of various ways. So we will see actually all those applications. So first of all, let's just see how or what is actually the GMO. So GMO, already I have mentioned that GMO stands for genetically modified organisms. So one bacteria within, suppose E. coli, inside the E. coli if we have inserted the rDNA, recombinant DNA, then that bacteria or that host will be also a genetically modified organism, because we have changed the genetic makeup of that organism. So that will be also a genetically modified organism. So GMO are more tolerant to the abiotic stresses like cold and drought. See, in India basically you can see various changed environmental terrain where we can see the extreme hot condition, extreme cold condition, then plain also we have, hilly areas also we have. So in all those areas we have to produce different types of crops, so that we can able to make that crop resistant to extreme cold or extreme hot, or in the drought also, as well as flood also. So that's why lots of plants, they have been manufactured in this way, so that they can be tolerant, they can survive in those extreme conditions. Like they can survive in, suppose in very less water, they can survive in extreme saline or extreme salty soil also, they can survive. So this way the plants are created, they are biotechnologically, they are modified so that they can withstand all those extremes, environmental extremes. Next one, it reduces the tolerance of the chemical pesticides. That means whenever we produce a GMO, genetically modified organism, in agriculture. So this actual GMO we are discussing here are in the field of agriculture, that whenever we form or we create one genetically modified plant or that seed or the fruit, then those fruits, they are actually formed, they are created without the use of any type of chemical pesticides or any type of chemical fertilizers. So without the use of chemical fertilizers or chemical pesticides, we can make that plant more and more helpful or more and more useful, so that it cannot give the different bad chemicals or different chemicals to the food chain, so that it can harm the other organisms. So that's why some concept have arisen, that is the organic farming. So in the organic farming also, the genetically modified organisms, they are grown without the use of all those chemical pesticides, because it is not only harming human but also the other organisms, both animals, plants and microbes, that decrease the soil fertility. It also cause the death of lots of other organisms too. Not only that pest, but also it kills the other organism which is not targeted. So this GMO, genetically modified organisms, they are actually used to decrease the uses of all those chemical fertilizers or chemical insecticides or chemical pesticides. Next one, it helped to reduce the post-harvest loss. See, post-harvest loss is very actually one serious problem, that whenever suppose a plant is grown in that specific season, only in that season that specific crop is grown by the farmers, and in that time the availability is so much more that that's why the cost get decreased. And after that, what happened, lots of these plants, they are just wasted because the production is over, so that all the people, they are consuming it also, and if they have to consume it, then the farmers will be in loss. So that's why we have also solved these problems by the use of biotechnology, that this plant can be produced, that means how much suppose kilograms or tons are required by this city, only that amount of crops will be they will be harvested, not more than that. And if it is more than that, then also that have to be consumed by, or it will be not at least go at lost by the farmers. The farmers will be not in loss due to this biotechnology. Next one, it increased the efficiency of the mineral uses by the plants. So these plants, they require the different type of elements, minerals like phosphorus, calcium, sulfate from the soil. So that will be also one application, that whenever the plant extracts those minerals from the soil, it do not completely decrease the soil fertility. This way the genetically modified organisms, GMO, are created. Next one, some examples are given. See, to enhance the nutritional value of the food. Suppose nowadays we have this type of some deficiency diseases due to different type of vitamins, because whenever it comes to the busy schedule of our life, we are not concentrating on the food value, rather we just consume it to decrease our hunger. So what happens whenever it comes to the nutritional quality, just by suppose one superfood, suppose from that we can extract different type of elements, then it will be a best product. But naturally this is not happening. Suppose rice is a source of carbohydrate, right? in that same rice also we have introduced some nutritional value, that is vitamin A precursor, that is introduced, and that make the rice golden rice, which is enriched with vitamin A, which is introduced into the rice plant, so that this rice plant can give not only the carbohydrate but vitamin A also. So this way we can able to decrease the vitamin A deficiency, which is very much common, and along with that we also can decrease the uses of the different types of food, because we have to purchase the food, we have to cook the food, and we have to eat it. If we get only that suppose specific that rice, that is the golden rice, then by this golden rice we can able to extract vitamin A also, carbohydrate also. So that is one very revolutionary discovery of the biotechnology, that in the food product we can get lot of minerals from the same product. That is one example, that is golden rice. We will discuss golden rice in detail, we will see all of it. So just see, this biotechnology can be used in different fields. We will see first of all in the field of agriculture, then we will see in the field of medicine, we will see lots of other areas. First of all, we will see biotechnology in the field of agriculture. That means in the agriculture, how biotechnology can be helpful. So first of all, this is to obtain a pest resistant plant by using BTG. So this is one very much important. That means without the use of any type of pesticides or insecticides, we can make the plant pest resistant, so that we should not give any type of pesticides on that plant, and the plant will grow without the use of any type of pesticides, and they can kill also the pest. So without harming the environment, it can be very much important that we can harm at least to the environment. So that's why this concept have arisen. One is very important, that is to obtain the pest resistant plant by using BTG. So see, what is this BTG? BTG is actually, this BT comes from one bacteria, that is Bacillus thuringiensis. So this Bacillus thuringiensis is one bacteria which have one type of gene, and that gene is known as cry gene. And this cry gene that undergo transcription and then translation. The cry gene undergo transcription, it will produce one mRNA, and that mRNA undergo translation to produce a protein, and that protein is known as the cry protein. Okay, so just see, cry gene, what I have written CRY, that C is small if it is the gene and the C is capital if it is the protein. Remember that only. So from the Bacillus thuringiensis, one gene is extracted, that is the cry gene. So this cry gene that undergo transcription and translation, and it produce one protein that is called as cry protein, which have one very interesting character. What is that? This cry protein is actually crystalline protein. Okay, it is normally a crystalline protein. But whenever this crystalline cry protein goes to any alkaline medium, it gets dissolved and it becomes toxic. Okay, so remember that this cry protein, normally it is crystalline, but whenever it reaches in the alkaline medium, then it turns toxic. In alkaline medium, it gets toxic. Okay, so this is the normal condition. Bacillus thuringiensis, that bacteria don't have any problem, because in case of Bacillus thuringiensis there is no alkaline medium, right? So the scientists have discovered that cry gene, and then the cry gene was inserted inside a plant, that is in India, there are various type of BT plants are available, like BT cotton, BT brinjal, BT soybean, various type of BT products, BT tobacco are available. From that, one plant we have taken, that is the cotton. Okay, so in that cotton sapling, that means in the cotton plant, okay, so this is the plant. In the cotton plant, the scientists have introduced that cry gene. Okay, right? So in that plant, the cry gene, what it is going to do? It undergo transcription translation. So whenever it undergo transcription and translation, what it is going to produce? The cry protein. But do the plant have any alkaline medium? No. So if the plant don't have any alkaline medium, alkaline environment, that means this cry protein is not going to harm the plant, right? So now this plant will grow, and in that large, long, huge cotton plant, everywhere, like in the seeds also, root also, stem, leaf, everywhere, they're going to have this cry protein, right? So they are going to have the cry protein. So in the plant, it is not at all harmful because it is in the crystalline state due to the absence of any type of alkaline medium. Now just see, to this plant, there are number of pests. That means this plant is taken by different type of coleopterans, dipterans, means the different type of flies, then different type of worms like cotton ball worm, then different type of beetles. So these are the pests of the cotton plant. So that pest, suppose pest larvae, okay, so this is one pest. If the pest larvae take the leaf, okay, take the leaf of that transgenic plant or GMO, same. So whenever this pest larvae take the leaf of that BT cotton plant, then inside the GI tract of the larvae, that cry protein enters. Okay, and we know that in case of the animals, we have the bile in our intestine, so that the arthropoda, they also have the alkaline medium in their intestine. So in that alkaline medium, what happens, that this cry protein dissolves. That means from the crystalline state, it gets liquidified, it gets dissolved. So suppose you just see, this is the intestine of the larva. Okay, so this intestine which have the cell epithelial tissue, okay, so they have the villi also, but suppose this is the intestine, right? Now whenever this cry protein comes in contact with the bile, that is the alkaline medium, what happened, that this crystalline cry protein, they start to get dissolved. It creates pores. In the intestinal wall. So whenever it creates the pores in the intestinal wall, what will happen? It will completely destroy the intestine. So if it destroy the intestine, that means it kills the larvae. So if we can able to restrict the larval stage, if there is no larva, how it will metamorphose to the adult, right? So at one period of time, the pest will be completely eradicated from that area, from that locality. That is the importance of this BT gene or that cry gene. So this cry gene, that is not harmful for the plant, but it is very, very much harmful for the pest. That means it is specific. That means if that plant is taken by some another organism, it is not going to harm that animal or that another organism. It is specific to only that pest. If the pest take it, then it will enter into the intestine. It completely destroy the intestinal wall and it create pores in the intestine and it kills the larva. And if it kills the larva, that means there will be no pest. So this makes the plant resistant to the pest. So that is the concept. To obtain the pest resistant plant by using BT gene, we can able to get the plant, pest resistant plant. So various type of pest resistant plant was there. In India in 2002, for the first time BT cotton was introduced by the scientist in India, and after that it was about to introduce into the brinjal, but later on there was lots of controversies came that it can harm not only the those larvae pest, or it can kill or it can harm human body also. That's why it was not introduced into that brinjal. But still now BT cotton, BT tobacco, also BT soybean, it is introduced this crygene, so that makes the plant pest resistant plant. So let's just see after that what is the next thing about this the same concept, that is how many different types of genes are especially to kill the different type of pests. So let's just see that. So just see, there are varieties of crygene which is specific to different types of pest. So once again I will tell you what actually this crygene works. Okay, so from the bacteria Bacillus thuringiensis, one gene, that is the crygene, that is extracted. This crygene is introduced into the plant. In the plant it transcribe and translate to form the cry protein. This cry protein is in the crystalline state, so that's why it is not able to harm the plant. Whenever it is taken by the different types of pest, suppose that plant leaf is taken by any type of pest, basically the arthropods, then inside the GI tract, this crystalline cry protein will enter. This crystalline cry protein, which is in the inactivated state, that is in the crystalline state, in the midgut, of this different pest like the corn borer, then Colorado beetle, corn rootworm, then the cotton bollworm, in those pests, in the midgut it starts to dissolve. Okay, so how it will dissolve? It will actually attach to the epithelial cells. Okay, create pores. And you know, whenever it create pores, what happened? Suppose the epithelial cells, it develop the pores, then obviously endosmosis will occur, the cell will start to swell, and if the cell swell at one period of time, the cell will burst, isn't that? So it create pores, then the cell swells, right? Then after the swelling, it ruptures. Okay, so if there is no midgut, no pest. So what will happen? This way, this whole cry gene will work. Just remember the difference between this and this. Okay, if it is this, CRY, C is small letter, then this is the gene, and if that C is capital, this will be the protein. Okay, now to see that how many different types of crygene is responsible for killing or destroying the various types of pests. So first of all, just see the first one, that is the protein encoded by the gene Cry1AC and Cry2AB. Remember, this is going to be very much important. It basically comes in the NEET. So in the NEET examination, this you will get at least one of the question, at least in your objective examination, you will get for sure 100%. So see, this protein encoded by the gene. From this also you can remember that the gene Cry1AC and Cry2AB kills the cotton bollworm. Remember that. Then after that, the second one, that is the protein encoded by the gene, that is Cry1AB, kills the corn borer. Next one, the third one, that is the protein encoded by the gene Cry3AB, kill the Colorado potato beetle. Next one, the food type of gene, that is the Cry3BB, kills the corn rootworm. So just remember, these are going to be very much important. So this is the first thing, that is to make that is the pest resistant plant. In the field of agriculture we have seen. After that, we will see a lot of pest resistant plant by another technique, that is the RNA interference technique. We will discuss in the next video. Till now, this is the first introduction of this chapter, that is biotechnology and its application, and the introduction of BT. So that's all about the video, introduction video. I hope you understand. Thank you.